Good morning, good afternoon, good evening, colleagues. We welcome you to the final session of the four-session TB Science 2020, with today's focus being on innovations in TB therapeutics. My name is Payam Nahid. I'm a professor of medicine at UCSF in San Francisco, California. I am pleased to serve as session co-chair with my colleague and renowned clinical trialist, Dr. Amina Jindani, professor of tuberculosis therapeutics at St. George's University in London. 2020 marks the 40-year anniversary of her seminal publication with Professor Denny Mitchison on a new methodology for measuring the early bactericidal activity of drugs in patients with pulmonary tuberculosis, so-called EBA studies. This type of study has remained an enduring approach still used today in drug development. I would like to invite Professor Jindani to make some introductory remarks, but before handing over, I also want to take the opportunity to express our collective thanks to the Bill and Melinda Gates Foundation on behalf of the Union, the TB Science Scientific Committee, and the chairs for their continuous and strong support of TB science. Amina? Oh, you're muted, Amina.
Thank you, Payam, for your kind introduction. Firstly, enormous congratulations for coming up with the first ever 4-month regimen for drug-susceptible tuberculosis. That is study 31-A5349 results, which were presented on Wednesday at the Union conference. Secondly, I'm really very honored to have been asked to welcome a really stellar group of speakers on subjects of completely innovative methods of assessing treatment of tuberculosis. The objective is to find methods of rapidly identifying short duration treatments. However, since Payam and his colleagues have already pipped us to the post, perhaps we should just pack up and go home. But let's just hang in for a bit. The WHO Global Tuberculosis Report 2019 has just been published, and the summary is: progress is being made as assessed by the global indicators for reductions in TB cases, deaths, and improved access to TB prevention care. However, increased financing is moving in the right direction. Nonetheless, the pace of progress worldwide and in most regions and in countries is not yet fast enough. That is their summary. Since the annual estimated incidence of mortality has stubbornly remained at 10 million and 2 million respectively, I'm rather puzzled to know how they arrive at these conclusions. The report does go on to say the fight against TB remains chronically underfunded, which is the one fact I can agree with wholeheartedly. Once upon a time, the treatment of tuberculosis was really easy. All the afflicted had to do was to line up before the king, and His Majesty would lay his hands upon them. Now, of course, it is far more complicated, which is why it is so gratifying to see TB therapeutics is experiencing a renaissance significance in different methods of evaluation. There is a desperate search for biomarkers which could move trials from phase 2 to phase 3 more rapidly. Recently, this includes something called estimans, but I'm still trying to get my head around that one. In this session, you will hear about bug drug evaluations, the challenges for optimizing host-directed therapies, and genomic discoveries for developing host-directed therapies from a distinguished group of scientists. I will leave Payam to tell you more about the session, but as a clinician, I would really like to see many more phase 3 trials for drug-susceptible TB underway. Thank you.
Thank you, Amina, for those comments and reminding us to keep the race going. There's a lot more ahead of us, and I hope some of the discussion today will be engaged in that space. I'd like to now proceed with the 3 invited presentations. First, we're pleased to have a presentation from Professor Gavin Churchyard, who's a specialist physician, internationally renowned for his contributions in TB research. Professor Churchyard is the founder and chief executive officer of the Oram Institute in South Africa. He holds honorary professorships at the University of Witwatersrand and University of Cape Town. Professor Churchyard is also the chair of the NIH ACTG Transformative Science Group for TB, chair of the NIH Cross-Network TB Vaccine Working Group, and co-chair of the ID93 therapeutic TB vaccine trial. He has extensive clinical trials experience and has conducted numerous TB and HIV vaccine trials. Dr. Churchyard will present on the potential opportunities of trial designs for TB therapeutic vaccines. Next, we're very pleased to have an invited presentation from Dr. Joshua Vasquez. Dr. Vasquez is a pulmonary and critical care physician and assistant professor at University of California, San Francisco in the Division of Experimental Medicine. His research focuses on the role of myeloid cells in lung disease related to TB and HIV. The Vasquez lab has pioneered techniques to study single cell reservoirs of HIV in tissues. Dr. Vasquez has applied these cutting-edge tissue-based technologies and high-dimensional analytics to understand how diverse tissue microenvironments impact populations of MTB. And he will present on new innovative tools for evaluating drug effect in granulomas and special microenvironments. And finally, we're pleased to have an invited presentation from Dr. Margarita Saraiva, who studies cellular and molecular mechanisms in immune responses to mycobacteria. Dr. Saraiva received a degree in biochemistry from the University of Porto, Portugal. Her PhD is in pathology from the University of Cambridge. And she currently serves as president of the Portuguese Society for Immunology and is a group leader at the Institute of Molecular and Cell Biology in Porto, Portugal, where her lab studies the molecular mechanisms underlying immune regulation using deregulated myelopoiesis and tuberculosis as disease models. She'll present on the diversity of MTB and the impact that has on clinical outcomes, as well as the challenges that it raises for the development of therapies, in particular host-directed therapies. So after our three invited presentations, we'll have Q&A. Following that, there'll be an oral abstract presentations for excellent submissions to TB Science, and then finally a panel discussion. Please do send all your questions throughout any and all of the presentations, and Amina and I will monitor. And I thank you very much, and I hope you enjoy the TB Science session today.
Thank you for inviting me to give this presentation on opportunities and risks using therapeutic TB vaccines to improve cures and shorten treatment for active disease. I have no conflicts of interest to disclose. I will briefly describe the aims of TB treatment, factors associated with poor treatment outcome, and then give an overview of therapeutic TB vaccines with respect to their characteristics, biological feasibility, opportunities, and risks, and trial design considerations. The goal of TB treatment is to safely achieve durable, non-relapsing cure, avert death. TB treatment, however, particularly for MDR TB, is long and toxic with drug-drug interactions with antiretroviral therapy. And globally, treatment outcomes for TB, TB-HIV, and MDR TB are suboptimal, with treatment outcomes for MDR TB being only 56%. An individual level meta-analysis of the 3 phase 3 fluoroquinolone trials has identified that patients with pulmonary TB may be classified using baseline characteristics into those at low, moderate, or high risk of poor treatment outcomes. The analysis suggests that persons with high-risk TB may require longer TB treatment, whereas those with low-risk TB may require a shorter duration of treatment. In addition to high smear grade and presence of cavities, the figure on the left shows that being HIV positive and increasing BMI are risk factors for poor treatment outcomes. The figure on the right illustrates that patients with high-risk TB, represented by the red line, have a far higher proportion of unfavorable outcomes over time than those with low or moderate risk TB. The first-line TB regimen for drug-susceptible TB is unforgiving of poor adherence. Poor adherence, mostly in the continuation phase, is a major driver of unfavorable treatment outcomes. The meta-analysis highlighted that patients with greater than 15% poor adherence had a substantially increased risk of poor treatment outcomes. Recurrent TB occurs in up to 8% of persons that successfully complete treatment for drug-susceptible TB with a first-line regimen. Recurrent TB constitutes between 5 and 30% of the TB burden and may be due to relapse or reinfection. In high-burden settings, recurrent TB is mostly due to reinfection, particularly after more than a year post-treatment. In Cape Town, the rate of TB following a previously treated episode of TB is 2 to 17 times higher than the background case notification rate, depending on the number of previous TB episodes and the treatment outcome of the previous TB episode. The rate of recurrent TB is substantially higher amongst persons who did not complete treatment for their last TB episode. Most TB patients are left with permanent, clinically significant lung function impairment due to fibrosis and bronchiectasis. The more severe the TB disease, the greater the degree of lung function impairment, as shown in the figure. Lung function impairment is associated with an increased risk of long-term death. A meta-analysis of studies evaluating the long-term consequences of TB have identified that in comparison to persons who have not had TB, the risk of death is increased approximately threefold post-TB despite cure. The mechanism of the increased risk of death are uncertain, but appear to reflect local and systemic effects of persistent lung inflammation. TB vaccination strategies include prevention of infection, prevention of disease, prevention of recurrence, and therapeutic TB vaccines. Our focus will be on the last two vaccine strategies. Therapeutic TB vaccines are given to persons during or at the end of TB treatment and seek to induce appropriate immune responses and aim to achieve both individual benefit and contribute towards population-level impact. Therapeutic TB vaccines will be targeted to patients with drug-susceptible or resistant TB, regardless of their HIV status, with the aim of improving cure rates, decreasing the rate of recurrence, and simplifying treatment by reducing the duration of treatment or reducing the number of drugs in the regimen. The evidence of biological feasibility comes from a number of areas. Epidemiologically, TB infection reduces the risk of subsequent TB infection and disease by up to 90%. Preclinical models provide evidence that TB vaccines given adjunctively with TB treatment may allow for treatment shortening and improved outcomes as illustrated in the mouse models for M. indicus prani shown in the top figure, where treatment alone with a standard first-line regimen sterilized cultures after 16 weeks, whereas treatment administered with the vaccine sterilized cultures after 12 weeks. Similarly, for the ID93 GLA-SC vaccine shown in the bottom figure, vaccination with a shorter duration of treatment was more effective than a longer course of treatment alone. Evidence from the recently published M72-AS01E clinical trial demonstrated that the vaccine administered to HIV-negative persons with latent TB infection reduced the risk of TB disease by half. A meta-analysis of adjunctive MVACI with MDR TB treatments suggests that therapeutic vaccines may improve outcomes, although most studies were small and of poor quality. Therapeutic TB vaccines may work through one of two mechanisms: host-directed therapy with the aim of modulating the immune response from a Th2 biased to a Th1 response, which will increase the efficacy of chemotherapy. These vaccines can be administered at the start of treatment or soon afterwards. Examples of these vaccines include those derived from non-tuberculous mycobacteria, such as M. vacci, obuensa, and M. manrescensis. Killing the bacilla by boosting the immune response, directed at persistent organisms with slow replication and reduced drug sensitivity by using vaccines that express latency-associated antigens. These vaccines should only be administered after significant reduction of the bacterial load with chemotherapy. Examples of these vaccines include M. indicus prani, Ruti, ID93, and H56. The pipeline of therapeutic TB vaccines currently in clinical development include whole cell, subunit, viral vectored, and live recombinant vaccines that are in phase 1, 2, and 3 trials. There are also two exciting vaccine candidates in preclinical development, one which is a viral vectored and the other a next-generation subunit vaccine that may also be used as therapeutic TB vaccines. I will now discuss the opportunities and risks for prevention of recurrence, improving cure, and simplifying treatment. Vaccination after successful TB treatment aims to reduce the rate of recurrence due to relapse and reinfection. From a product development perspective, prevention of recurrence trials can be used to demonstrate proof of concept prior to conducting larger prevention of disease trials. A TB vaccine to prevent recurrence may be of greater benefit in drug-susceptible TB, where the risk of recurrence is greater than the risk of treatment failure. However, with recent advances in treating MDR TB, prevention of recurrence vaccines may also be of benefit in patients with MDR TB. As treatment will have massively reduced the bacterial load, the risk of vaccine-induced exacerbation of disease is likely to be low. Potential TB vaccines for prevention of recurrence include the ID93 GLA SC vaccine, and the safety and immunogenicity of a 2 or 3 dose regimen of ID93 has been evaluated in a phase 2A trial in HIV uninfected adults with drug-susceptible pulmonary TB. The safety and efficacy of H56 IC31 is currently being evaluated in patients with drug-susceptible pulmonary TB, and the safety and immunogenicity of the M72AS01E vaccine was evaluated in patients with drug-susceptible pulmonary TB, but was stopped early due to large injection site reactions. And the efficacy of VPM1002, a recombinant BCG vaccine, in preventing recurrent TB is being evaluated in patients with pulmonary TB. TB vaccines administered during treatment may improve the proportion cured and reduce the risk of recurrent TB disease. Therapeutic vaccines may be particularly beneficial amongst patients with suboptimal adherence and hard-to-treat TB. Therapeutic TB vaccines may also reduce the risk of developing drug resistance. The benefits, however, may potentially be offset by a higher risk of adverse reactions, including Cox phenomenon. Overlapping toxicities may occur with TB treatment, particularly if given during the intensive phase of treatment and in patients initiating antiretroviral therapy. Potential TB vaccines in development that may be used to improve cure include MVACI. A meta-analysis of 54 trials, including studies outside of China, suggest improved treatment outcomes. A large phase 3 trial is ongoing, and of note, MVACI is already approved as adjunctive therapy for TB in China. M. indicus prani showed a modest improvement in culture conversion, but not in rate of recurrence. The safety and immunogenicity of RUTI is currently being evaluated in patients with MDR-TB. H56 IC31 vaccine with a cyclooxygenase inhibitor is being evaluated in drug-susceptible TB. A planned trial of ID93 GLASC will evaluate safety, immunogenicity, and efficacy in patients with moderate and hard-to-treat drug-susceptible pulmonary TB. A vaccine that has been shown to improve cure may potentially be used to simplify treatment with a shorter novel regimen or a more compact regimen with fewer drugs. The molecular changes resulting in drug resistance are unlikely to result in changes to antigenic proteins included in vaccine candidates. Therefore, vaccines capable of preventing recurrence of drug-susceptible TB are likely to have similar activity against drug-resistant TB. Programmatically, however, patients with hard-to-treat drug-susceptible pulmonary TB or MDR-TB may derive greater benefit from therapeutic vaccines due to poor treatment outcomes. Patients with MDR-TB that fail treatment or die tend to have a lower probability of sputum culture conversion compared to those who successfully complete treatment. As shown in the figure, the majority of patients with MDR-TB that successfully complete treatment convert their cultures within 2 months, whereas only 20% of those who fail treatment or die converted their cultures by 2 months. So what can be done to increase the proportion that convert their cultures and decrease the risk of treatment failure and death? The World Health Organization developed regimen characteristics for novel MDR-TB treatment regimens. Although numerous regimen characteristics were identified, efficacy was thought to be the most important regimen characteristic, and even small increases in efficacy can have substantial benefit. Modeling suggests that if only efficacy of a MDR-TB regimen is improved, shown by the red dashed line, by using more potent drugs or possibly by using adjunctive therapeutic TB vaccines, this may have considerable impact on TB-related incidence and mortality. Other regimen improvements, however, may still have important benefits for resource allocation and outcomes, such as patient quality of life. I will now discuss trial design issues related to evaluating safety and efficacy and propose some exploratory outcomes. This cartoon shows possible vaccination time points for therapeutic and prevention of recurrence TB vaccines. Vaccination to or at the end of treatment is appropriate for trials of prevention of recurrent TB. Vaccination at the end of the intensive phase of treatment is proposed for trials evaluating TB vaccines for cure and treatment shortening. However, due to safety concerns, it would be unwise to immediately start assessing therapeutic TB vaccines administered at the end of the intensive phase. Safety should ideally be evaluated using a timed escalation of vaccinations approach. Illustrated in the figure is an example of a timed de-escalation approach being considered to evaluate safety of a therapeutic TB vaccine where enrollment of each group requires demonstrating safety in the previous group. As the adverse event rate amongst placebo recipients is greater than 0% and increases with proximity to the treatment start, pause and stop criteria should be applied based on a difference in rate between the vaccine and placebo groups. As already mentioned, adherence is a strong predictor of poor treatment outcomes. Adherence to TB treatment in routine programs is poorer than that achieved in clinical trials. From a clinical trial perspective, the sample size required will be strongly influenced by the overall adherence to TB treatment. A novel approach would be to allow the routine program to remain responsible for TB treatment, and the research team should be responsible for vaccinations and evaluating safety, immunogenicity, and efficacy. Using this approach, the required sample size to show efficacy could be reduced. Shown in the figure is a model simulation of the proportion with unfavorable outcomes under program conditions compared to treatment under program conditions with the vaccine. Efficacy could also be evaluated in a smaller phase 2A trial using proportions culture conversion and ×2 culture conversion. Therapeutic TB vaccines must be safe and effective in people living with HIV who are on antiretroviral therapy or those that are ART naive. Limited evidence suggests that immunogenicity may be lower in people living with HIV than that observed in people without HIV. There are numerous complexities with including people with HIV in therapeutic TB vaccine trials, which includes when to include people living with HIV in TB vaccine trials, what CD4 count eligibility criteria to use, and in the proof of concept trials, should the number of people living with HIV be capped in order to maximize the chance of showing efficacy. TB-associated lung function impairment persists despite treatment, but may be reduced with host-directed therapy as shown in the figure on the left. An RNA gene signature has been shown to increase with TB disease progression and decrease with TB treatment as shown in the figure on the right. Exploratory endpoints which may be considered include use of lung function tests and other measures of respiratory function, such as a respiratory questionnaire and the 6-minute walk test, and RNA gene signatures. Finally, I recommend consulting the World Health Organization Preferred Product Characteristics for Therapeutic TB Vaccines, which has recently been published and includes some characteristics we have already discussed, such as indication, target population, outcome measures and efficacy, immunogenicity and safety, and others such as duration of protection from recurrence, programmatic suitability, and the value proposition. In conclusion, globally, treatment outcomes for all forms of TB are suboptimal. The opportunity exists to develop TB vaccines that may reduce the risk of recurrence and post-TB morbidity, improve cure, and simplify TB treatment. Novel trial designs may be required to evaluate TB vaccines to improve cure and shorten therapy. Therapeutic TB vaccines may contribute to population-level impact. I would like to thank those that have contributed to the concepts proposed in this presentation. Thank you.
I have no conflicts of interest report. Hello, I'm a pulmonary and critical care physician from San Francisco. I conduct translational research focused on host pathogen interactions in TB. Today, I will talk about the physiologic state of Mycobacterium tuberculosis within tissue microenvironments. When we were treating TB, we were treating many different populations of bacteria that occupy different tissues. For example, the populations of bacteria residing within the airway, cellular lesions, and the caseum are exposed to different environmental stressors. This led Dr. Mitchison to hypothesize that those bacteria also grow at different rates and therefore respond differently to specific drugs. In other words, we lack a pharmacodynamic marker that defines the efficacy of individual drugs on these distinct populations of bacteria within an infected host. My talk today has 3 parts. In part 1, we will evaluate differences between the mycobacterial populations in the caseum, cellular lesions, and bronchoalveolar lavage, or BAL, from the airway. In part 2, we will look at differences between the mycobacterial populations in the granulomatous myeloid cell layer and the central caseum. Then in part 3, we will look at heterogeneity within populations of bacilli in both the myeloid cell layer and the caseum. For the work that I will describe today, we use the Kramnik mouse. These mice were infected with Erdmann strain by low-dose aerosol for 10 weeks. Mice were either left untreated or dosed daily by oral gavage with a single antibiotic for 2.5 weeks. Antibiotics used in this study were isoniazid and rifampin at 2 different doses. Tissues from these mice were separated and either processed to extract total or bulk RNA for quantitative PCR or left intact and prepared for histological analysis, including in situ hybridization. Tissues for bulk RNA analysis included bronchoalveolar lavage from the airway, caseum, and cellular lesions. In situ hybridization was performed on intact caseous lesions, followed by high-resolution imaging and quantitative image analysis, which I will describe in more detail shortly. The work that I'll share with you today is based on the concept of a pharmacodynamic marker called the ribosomal synthesis ratio. In mycobacteria, the entire ribosomal operon containing 16S, 23S, and 5S is transcribed on a single precursor transcript. This includes stable, mature, structural ribosomal sequences that are separated by unstable, non-coding spacer sequences that are rapidly excised integrated. The ribosomal synthesis ratio is based on the idea that the spacer is a short-lived byproduct in the production of ribosomes. Therefore, the abundance of spacer sequence indicates ribosomal synthesis, and the abundance of unstable spacer sequence relative to stable structural ribosomal RNA provides a simple marker of a fundamental physiologic parameter. We have found that a drug's capacity to suppress ribosomal synthesis correlates with that drug's sterilizing activity. That is, the more potent a drug or regimen is in shortening treatment, the more it suppresses the ribosomal synthesis ratio, which allows us to use this measurement as a pharmacodynamic marker to assess treatment responses in multiple models in human disease. Now, in part one, I will show you how we use this ratio to confirm that the bacterial populations found within different tissues are distinct. Specifically, they have quite different rates of ribosomal RNA synthesis. Here, the x-axis shows different tissue types. The y-axis shows the ribosomal synthesis ratio on a log scale. Dots represent individual tissue samples from a total of 45 mice. The horizontal bar indicates the median value for each tissue. Relative to airway, the ribosomal synthesis ratio was twofold lower in cellular lesions and tenfold lower in caesium. Since ribosomal synthesis is directly linked with bacterial replication, these findings are consistent with a rapidly replicating population in the oxygen-rich airway and a slowly replicating population in the hypoxic caesium. The reason this is so important is that we have long hypothesized that drugs operate differently on different populations. The ribosomal synthesis ratio enables us to measure this directly. To illustrate the concept, I will show you two drugs that affect these populations differently, rifampin and isoniazid. Here is a reference. I am showing the same untreated control. Here is a reference. I'm showing the same untreated control for cellular lesions in the caesium shown on the last slide. Prior to treatment, mycobacteria and caesium are synthesizing ribosomal RNA more slowly. Rifampin suppresses the ribosomal synthesis ratio in the populations of bacteria from both the caesium and cellular lesions. The effect of isoniazid was different. Isoniazid suppresses ribosomal synthesis in cellular lesions, although not as much as rifampin. However, isoniazid had no effect in the caesium. This is not because isoniazid does not penetrate the caesium. At this dose of isoniazid, its concentration in caesium reaches levels well above the minimal inhibitory concentration. Rather, this is consistent with a well-established observation that isoniazid is most effective against rapidly replicating mycobacterial populations. These results establish the concept that we can use the ribosomal synthesis ratio to evaluate the effect of drugs on the bacterial populations found within key tissues. Now in part two, we dig a layer deeper and explore differences in mycobacterial populations occupying different regions or microanatomical locations within a given lesion. In the mid-1940s, Dr. Georges Canetti predicted that bacterial populations within tuberculosis lesions exist in diverse physiologic states that drive the clinical course of TB and the response to therapy. The Kramnik mouse is ideal to study this hypothesis since it develops heterogeneous lesions that compare to those produced in human infection. The type 1 or caseous lesion depicted here resembles the classic TB granuloma in humans. It is encapsulated within a fibrous ring with an area of caesium at its center that is low in oxygen and nutrients. Between these two regions is a layer of myeloid cells that includes highly vacuolated foamy macrophages and an adjacent layer of neutrophils. Given the differences in environmental stressors between these regions, Dr. Mitchison would have predicted that the local bacterial populations would be in very different growth states. In other words, we use the abundance of premature ribosomal RNA in individual bacteria as a bacterial vital sign that provides a pharmacodynamic marker that can be used to determine the physiologic state of bacteria within lesional microenvironments. Now I will show you an example of duplex in situ hybridization performed on a single section of tissue from a caseous lesion of an untreated Kramnik mouse. The H&E section on the left provides orientation. We can see relatively uniform staining of mycobacteria harboring mature ribosomal RNA in green. Staining for unstable premature RNA in red is heterogeneous across the lesion, with more abundance and intensity around the periphery in the area corresponding to the myeloid cell layer. This indicates more new ribosomal synthesis in the bacilli in that region compared to the nutrient-poor caseum, where premature RNA signals are fewer and less intense, indicating less ribosomal synthesis. We will now look more closely at these two regions. In these higher magnification images, we can begin to identify the filamentous shapes of individual bacilli with high and low signal intensities for premature ribosomal RNA, co-localizing with more abundant and mature ribosomal RNA. The myeloid cell layer on the left is divided into the region of foam cells on the left of the dashed line and neutrophils on the right. The white arrows point to foam cells that harbor intracellular bacteria that are seen congregating around host nuclei in blue. On the right, the caseum also contains mycobacteria with various levels of staining intensity for premature ribosomal RNA, but with fewer bacilli that exist at high intensities compared to the myeloid cell layer. With further magnification of the myeloid cell layer and using a green stain now to identify the foam cells, we can clearly distinguish filamentous bacteria stained for premature ribosomal RNA in red. Many of these bacilli are seen within and around a foam cell that is directly interacting with the T cell in whites at a synapse labeled in magenta. I love these particular images so much that I wanted to include one more high magnification image from the myeloid cell layer that identifies a foam cell filled with intracellular bacilli, stained by duplex in situ hybridization, and then displayed separately by channel. This image allows me to illustrate an important point about the analysis of the work that I will show you next. Unlike PCR-based analysis of bacterial RNA extracted in bulk from host tissues, in situ hybridization allows us to identify the population of bacilli by targeting the more abundant, stable, mature transcript in green, than to estimate ribosomal synthesis within individual bacilli. We measure the intensity of staining for the unstable premature transcript. Therefore, when we measure the average intensity of premature ribosomal RNA signals, we are measuring the average for the entire population of bacilli that are positive for the mature transcript, whether or not they are also positive for the premature transcript. Now I will show you how we analyze these signals. This is a spatial heat map of pre-ribosomal RNA signal intensities from over 150,000 individual bacilli identified within a single caseous lesion. We use this heat map to localize quantified signals from individual bacteria. To quantify these signals, we use machine learning to identify individual bacilli based on physical and optical properties, and then generate the spatial heat map using coordinates of their location. This analysis colors the bacteria according to the intensity of staining for premature ribosomal RNA, where brighter colors indicate more intense signals that reflect more synthesis of new ribosomes. The green line separates the caseum from the myeloid cell layer. Similar to the previous images, we can see that brighter pre-ribosomal RNA signals can be found within the myeloid cell layer at the periphery of the lesion. After quantitative image analysis, we found that the intensity of pre-ribosomal RNA is 3.5 times higher in the myeloid cuff, or in the myeloid cell layer, compared to the caseum, indicating a higher rate of bacterial growth in that region. These results are in line with the idea that slower growing bacteria are found within the oxygen-low and nutrient-poor caseum. This demonstrates that using in situ hybridization and quantitative image analysis for pre-ribosomal RNA, we can visualize, quantify, and localize physiologically diverse populations of mycobacteria. Dr. Mitchison hypothesized that faster growing bacteria would be more efficiently killed by isoniazid, and drugs like rifampin would be particularly efficient at killing slower growing bacteria like those in the caseum. We confirmed this hypothesis using our in situ hybridization-based approach to evaluate caseous lesions from infected Kramnik mice. In this analysis, we focused on the central caseum. Here, the x-axis shows each treatment type and the y-axis shows the percent decrease in the average pre-ribosomal RNA signal intensities among the population of bacilli within each lesion. Compared to the untreated control represented by the blue box, relative to the untreated control, isoniazid decreased the average intensity of pre-ribosomal RNA signals by just 2%. In contrast, rifampin at 10 or 30 milligrams per kilo decreased signals more substantially and in a dose-dependent manner. This is in line with earlier analysis using a PCR-based approach to estimate mycobacterial ribosomal synthesis in these tissues. It also supports Dr. Mitchison's hypothesis, since at this dose of isoniazid, its concentration in the caseum is well above the minimum inhibitory concentration. Now, as we look at the myeloid cell layer, recall the intense staining for premature ribosomal RNA we observed in this region. Based on that observation, we would expect isoniazid and rifampin to effectively suppress pre-ribosomal RNA signals in this region. You can see that in the layer of myeloid cells, which is directly adjacent to the caseum, each treatment effectively suppressed premature ribosomal RNA signals. This is consistent with previous observations that isoniazid is most effective against rapidly replicating mycobacteria. Importantly, these findings support the concept that tuberculous lesions are made up of distinct microenvironments that support very different populations of mycobacteria with diverse physiologic states that respond differently to therapy, which establishes the important role for tissue-based single bacillary analysis of mycobacterial ribosomal synthesis to evaluate the effect of drugs on the bacterial populations found within key tissue microenvironments. The work of John McKinney indicates that there is likely heterogeneity within populations of bacteria found within distinct microenvironments. We will use our tissue-based approach to look more closely at the physiologic state of these populations. Looking more closely at this spatial heat map of premature ribosomal RNA signals from individual bacilli, we can see different signals of different intensities within each region. The violin plot on the right shows the distribution of these signals among the population of over 150,000 bacilli. Signals from the population found in the myeloid cell layer are in orange on the left, and those from the caseum are on the right in green. The vertical axis indicates the intensity of premature ribosomal RNA signals. The distribution of signals indicates heterogeneity within the population with a range of physiologic states in both regions. To further evaluate heterogeneity of these populations, we incorporated data from in situ hybridization with over 90 other morphological and optical parameters captured for each bacilli into a high-dimensional analysis using unbiased clustering and principal component analysis. Here, the principal components are presented on the X and Y axis, which represents a unitless linear measure combining these variables, as well as the staining intensity for mature and premature ribosomal RNA. This analysis identified over 29 morphological and physiologically distinct populations of bacteria. Next, to evaluate the impact of drug therapy on these populations, we repeated this analysis on bacilli identified from lesions of animals treated with rifampin at 30 milligrams per kilo. And we found just 5 dominant populations of bacilli. Interestingly, 3 of those populations were not found in the untreated granuloma at all, and the other 2 made up less than 1% of the population. These populations were defined by a smaller and more ovid or less filamentous appearance, and the pre-ribosomal RNA signal intensities range from medium to low. Since lower ribosomal synthesis after treatment with rifampin was associated with decreased heterogeneity, we reasoned that differences in heterogeneity would also be seen in regions of the granuloma exhibiting differences in bacterial ribosomal synthesis. We found that among the 26 distinct populations of mycobacteria observed within the lesion from an untreated animal, only 21 were represented in the caseum, while 25 were found in the myeloid cell layer. Next, we evaluated whether isoniazid and rifampin differed in their impact on the breadth and distribution of mycobacterial heterogeneity. As a guide, I have included spatial heat maps for lesions from each of the treatments. Overall, we found that the heterogeneity of the mycobacterial populations decreased as the ability of a given treatment to suppress ribosomal synthesis increased. Looking more closely at the distribution of the remaining population, rifampin demonstrated a relatively uniform. Rifampin demonstrated relatively uniform populations between the caseum and the myeloid cell layer. Treatment with isoniazid, on the other hand, demonstrated more heterogeneity in the caseum compared to the myeloid cell layer. This observation is perhaps in keeping with the minimal impact of isoniazid we observed on mycobacterial ribosomal synthesis, although the markedly lower breadth of heterogeneity seen in the caseum after treatment with isoniazid is curious and warrants further evaluation. Today I discuss two tools based on the concepts of the ribosomal synthesis ratio to help us understand the physiologic state of mycobacteria in tissues. First, we used a PCR-based approach to estimate mycobacterial ribosomal synthesis occurring in tissues and found higher levels in the airway and cellular lesions compared to the caseum, which corresponds to slower growing populations previously predicted in this nutrient-poor environment. Next, by using duplex in situ hybridization to gain resolution of the physiologic state of bacteria at that level, we further confirmed a population in the caseum displaying low physiologic activity and also identified a physiologically active population that included intracellular bacilli in the adjacent myeloid cell layer. Third, our results uncover a spectrum of physiologically and morphologically diverse mycobacterial populations found in distinct lesional microenvironments and that support the long-held notion that slower growing bacteria within the caseum respond poorly to isoniazid compared to rifampin. Thus far, I have focused on the application of our work to resolve long-standing fundamental questions about the state of mycobacteria in tissues and the response to established drug therapies. However, our work provides a new approach to defining pharmacodynamic responses in tissues, which has important implications for TB cure. I'd like to acknowledge the efforts of those in my lab, as well as those from the University of Colorado, especially Nicholas Walter and the team from Colorado State University, including Gregory Robertson. Thank you for your time.
Okay, so I'd like to start by thanking the organizers for inviting me to speak at the TB Science 2020. And I'll speak about the diversity of Mycobacterium tuberculosis and the clinical outcomes of the disease. I think we all agree that tuberculosis is an heterogeneous disease, and we also probably all agree that the image is not as simple as it is presented here, because we have a lot of intermediate status in between the different phases that form different spectrums. And one of the spectrums that we can consider is the spectrum of TB severity that can vary from mild to moderate to severe. So, in my lab, in the last few years, we have been studying this spectrum of severity, and we are particularly interested in which factors modulate it. Of course, this is a central piece for this is the immune response, but the immune response itself depends on the interplay between the pathogen, the host, and extrinsic factors. So it's not a very simple question to address. And we decided to start focusing on the pathogen. Okay, so why did we think the pathogen would be a good option? On one hand, we know that Mycobacterium tuberculosis is a diverse bacteria. It follows a phylogeographic distribution similar to the human population. We also know that there are different links reported between the different lineages of the TB-causing bacteria and certain clinical features of the disease. And furthermore, our lab and others in the world have shown some relation between diversity of Mycobacterium tuberculosis and the triggered immune response. For example, by differential recognition of the bacteria by toll-like receptors. So with this in mind, we designed an experiment or a question to address whether the natural diversity of the Mycobacterium tuberculosis isolates could in fact influence the immune response towards a certain TB presentation. And what we did was to collect clinical isolates from patients with different TB severities, and then to study the interaction of these isolates with immune cells and see if there was a relation between the response triggered and the severity of TB in first place. So we did this in a cohort of 681 pulmonary TB patients in Porto, which we stratified in those that had no other diseases or that had several comorbidities. We looked at the ancestry of our population and found that it was rooted in the European, in the Portuguese and European ancestry expected, so a very homogeneous population in this aspect. And then we designed a clinical decision tree to stratify the severity of TB into mild, moderate, or severe, which was based both in local and systemic parameters. And we found this distribution that is shown here. And basically, when we looked at different host factors, like sex, the patient age, or the time of symptoms, we didn't find any particular statistic association between disease severity and these parameters. So the host population seemed to be quite homogeneous and yet showing different severity of disease. So we next decided to look at the bacteria population. And here we also had a very homogeneous situation with a very large predominance of lineage 4, as expected from a country in Western Europe. And within lineage 4, we had the majority of LAM, Latin American and Mediterranean cases. And what is interesting is that as we progressed from mild to severe TB, we did see a decrease in the LAM incidence, but this did not explain the entire situation. So there had to be other factors, possibly in the pathogen, in addition to the lineage. So with this part of the study, we eliminated promoters of severe TB in the host, like non-TB precipitators, and we found that our host ancestry and bacteria ancestry are actually in symmetry as expected in this part of the world. And so we decided to start addressing whether there could be a bacterial manipulation of the immune response. So for this, we selected 10 isolates from mild TB patients, 10 from moderate, and 10 from severe, and we infected PBMCs from IgG positive individuals, so completely unrelated donors. And as you can appreciate here, as we, and for all cytokines tested, as we progress from mild to moderate or severe TB, the cellular response was lower. So the pathogen was indeed severe TB-associated pathogens were induced lower cellular responses, and this was completely due to the pathogen, because across the different donors we always saw the same differences, so independently of the genetics of the host. So what is the underlying mechanism? And to address this, we started by analyzing the whole genome of the TB-causing bacteria. And we compared low cytokine inducers with high cytokine inducers. And our first unbiased approach did not show any promising SNP candidates to separate these two groups. So we decided to go for a biased approach, and we looked into a very important virulence factor of Mycobacterium tuberculosis, which is also linked to the manipulation of the immune response, and that is the ESX1 secretion system. And here, indeed, we found that strains that induced low cytokine responses were accumulating mutations either in components or in regulators of the ESX1 expression system, which suggested that direct manipulation of the macrophage response could be in place. So we selected 2 isolates, one associated with severe TB and a low cytokine inducer, and another one from a mild TB patient and a high cytokine inducer. And again, as I showed a minute ago, IgG-positive unrelated cells responded a lot more to the potent inducer. This was also the case in IgG-negative. It was also the case in monocytes, in a cell line, THP1, and even in a different species, because we see the same thing in mouse bone marrow-derived macrophages. So we next performed RNA-seq analysis of infected macrophages, and what we saw, what was striking, is that the severe TB isolate that induced low cytokine responses did induce a lot less changes in the macrophage than the mild TB-associated isolate. So it seemed like the macrophage was not seeing this bacteria, this pathogen, as well as it was seeing the mild TB-associated one. And when we did the pathway analysis, it completely reflected this hypothesis, because whereas with the mild TB strong cytokine inducer, we had a lot of pathways linked to the immune response being triggered. With the low inducer severe TB1, that was not the case, and each pathway actually contained very few genes. So we thought that the fact that the low cytokine inducer is accumulating mutations in the ESX-mediated secretion system, and that is not triggering the macrophage to the same extent, might actually be correlated. And the first thing, so I just have the IL-1 beta graph here to remind you, the first thing we tested is, could this be a question of dose? If we increase the dose of the bacteria, can we recover IL-1? And the answer is no. So it's not a question of dose. The second thing we addressed was whether this was a question of actively inhibiting IL-1 secretion. And in fact, it's not, because in several competition experiments, we could never compete with the low cytokine inducer, the effect of the high cytokine inducer. So it was not an active process of inhibition. So next, we thought, okay, if this is related to ESX1 mediated secretion system, we need metabolically active bacteria. And in fact, if we block the metabolism of the bacteria with a high dose of rifampicin, the high IL-1 beta inducing bacteria is no longer able to do so. So it seems an active process that depends on an active bacteria. And one thing that is important to mention is that the transcription of the IL-1 beta gene was actually very similar to these two bacteria, suggesting that the differences do not occur at the initial level, but instead at the processing level. And the processing of IL-1 beta is linked to the inflammasome. So we measured the inflammasome activity, and it found it was in fact highly triggered by the high cytokine inducer bacteria, but not by the low. And if we blocked the inflammasome activity, the IL-1 beta disappeared. So this now starts to put the puzzle together, showing that metabolically active bacteria can differ on their ability to export molecules to the cytosol and to trigger, for example, the inflammasome. And this is also seen in another cytosolic surveillance system, that is CGAS, where the strong cytokine inducer had a very potent CGAS activation across different genes. And again, if we take a hallmark gene that is interferon beta and block CGAS, down goes the regulation of interferon beta. So you can find all the details and all the experiments in our recent publication, but what we did here was to use clinical data, to start with clinical data, to select relevant isolates of MTB, and then to combine whole genome analysis with a lot of in vitro infections and mechanistic studies to show that the bacteria isolated from patients with different severities can in fact modulate macrophage differently. And this is down to the cytosolic intracellular surveillance pathways, and it occurs across different donors and even across different species. So this kind of makes our point that there is a link between diversity of the bacteria, the immune response, and the severity of TB. But one question I get a lot is what happens in vivo, because of course there you have a lot more factors combined. to get the full immune response. So what I'm going to show now is unpublished data, and we are starting to address this question by infecting mice with the different MTB isolates. And we are finding that if we infect wild-type mice with a low dose of bacteria, we have a higher bacterial burden in case of the severe TB in the lung, so suggesting a more severe infection, in fact, also in the mouse. This is also seen if a high dose is used, and most interestingly, even in the absence of interferon gamma, a key protective molecule in TB, we are still seeing this difference. And this to us is suggesting that the initial differences in virulence between these two strains are actually fixing the odds of having a more or less severe TB, even in the absence of key molecules like gamma. And recent data are suggesting or are placing the IL-1 beta IL-1 receptor pair as a key molecule driving the early establishment of infection. So because we have bacteria that modulate IL-1, we think that our phenotype and our mechanism might be linked to these initial steps that are occurring in the lung. So we are also seeing this in vivo, and because of that, we believe that using Mycobacterium tuberculosis diversity as a tool to study the progression of disease, the dynamics of the immune response, and the pathology that is developing is actually a good strategy to define new mechanisms of TB severity that we can then explore as therapies. And we are pursuing these studies also in view of a recent paper from Anil Gara's lab in which we collaborated, and that shows that the mouse model can mimic the immune response seen in active TB patients. So combining all these, we are confident that we can uncover and test new strategies to modulate TB based on TB diversity. So I hope I have convinced you or that we can discuss that new strategies to modulate TB may have to evolve from host-directed therapies to host pathogen-directed therapies, because at least for a series of hosts, the diversity of Mycobacterium tuberculosis itself may be enough and sufficient to modulate immune response, and this may lead to the need to fine-tune the directed therapy to also encompass this bacterial manipulation. Of course, for extreme hosts with very low or very high immune responses, that might not be the case. So I'd like to finish by thanking the people who did this work, and I have 3 people in the lab that were really instrumental, and that's Jeremy Souza, Baltazar Ka, and Helder Bastos. We have other collaborators, both internal, national, and international, and I would also like to thank the funding agencies and you for your attention.
Wonderful. Thank you, Margarita, for that outstanding talk, and also to our other invited presenters, Gavin and Josh. Very thought-provoking and actually complementary discussions. We're going to now transition to our Q&A session. And I will, we've received a few questions on the platform. I would like to actually just remind everyone that on the conference platform where you're watching the presentations, there is a box that you can click that allows you to type in questions, and we welcome them during this session, and Amina and I will moderate. I want to start first with a question for Gavin, if I may, and then we'll transition to those that are coming in the chat, in the question box. Gavin, you know, very, for me, convincing arguments for the value of a vaccine applied in a therapeutic fashion. I wondered whether you had a thought about whether it would be possible in the trial designs or otherwise to distinguish protection that is from relapse versus new infections in endemic settings,
And perhaps it's not so simple to address that question, and maybe it doesn't really matter. But which do you hypothesize would have the biggest impact? And please do expand on this in other ways as well, if you'd like.
Thanks very much, Pam. So we should be able to differentiate between recurrence due to relapse versus reinfection, because with a therapeutic vaccine, we will be able to, in the clinical trial, collect the isolate from the patient at diagnosis. So we should have that, the infecting strain, and then should the patient develop TB post-TB treatment, again, we would then do genotyping and sequencing of that strain and be able to compare to see whether this was a relapse versus reinfection. Hopefully, the vaccine responses should protect from both. However, epidemiologically, we know that most recurrence, particularly the further out you go from having completed treatment, is due to reinfection. And so the greatest contribution of the vaccine will be to preventing recurrence due to reinfection.
Thanks, Gavin. I appreciate that. We had a question from the audience. It looks like you typed an answer, but maybe you could expand further. The question is, do you have any thoughts on why large injection site reactions with M72 in people with drug-susceptible pulmonary TB were seen, but not in the TB prevention studies?
Thanks, Pam. So the injection site reactions, or local reactions, occur commonly with subunit vaccines. And these reactions are due to the local immune response to the vaccine antigens. And why M72ASO1E resulted in these very large local reactions, I think if I remember correctly, it was in 2 out of 7 patients. And other subunit vaccines that have also been given at the end of TB treatment, such as the H56 and ID93, is not entirely clear, but the one difference may be because of the adjuvants used. ID93 actually has 4 antigens that it includes in the vaccine, where the M72 has 2, but the adjuvants may be different enough that we're seeing this difference in the local reactions. We know that when we treat TB patients, but actually uninfected TB patients, so they're not on antiretroviral therapies, of course, that you can, it is observed and well described, get paradoxical immune reconstitution syndromes, which is really a local reaction to antigenic load that occurs. And a vaccine may also induce a local reaction to antigen where it occurs in the body, and particularly with disease, but much less likely with infection, because the bacterial load and antigenic load will be so much lower. And so, although it's a possibility, it has rarely ever been described. But you would not expect to see an injection site reaction when giving treatment of infection.
And can I just stay with you once more? Do you have any thoughts around the recent publication around just BCG vaccination and protection against respiratory viruses? There's a lot of discussion around that for COVID-19 as well. Could you anticipate any more expanded value to therapeutic vaccine beyond TB relapse?
Yes, so that's obviously very topical at the moment, and there's been lots of discussion around the potential contribution of these, particularly it's the wholesale vaccines that may, through the induction of trained immunity, have a response beyond reducing the risk of TB. And so there is clear evidence that a BCG may reduce the risk of viral infections and given to diverse populations, but more recently the publication in elderly suggests that it reduces the risk of viral infections. We know that BCG also is used for bladder cancer, for instance. So this whole issue around the potential role of trained immunity in protecting against infection.
There's been a lot of ecological papers being published. The early papers showed potential benefit of BCG. The more later ones do not show benefit, but there are 13 trials of BCG for the protection against COVID, for instance. And some of those trials will be reporting out fairly shortly. And so we'll then see what the potential is of BCG and other whole cell vaccines in reducing the risk of COVID-19.
Many thanks, Gavin. Very exciting and interesting hypotheses to consider. I'd like to move to Josh. We have some very nice questions coming in for you, Josh. David Lewinson asks, or states, beautiful imaging. Do you find evidence for bacteria that is not in a granuloma airways? Have you looked early after infection? I wonder if you'd like to comment on that.
Yeah, thank you very much for the question. We have not looked at different time points after infection. We do definitely see bacteria outside of the granuloma. We are able to see bacteria within alveoli and within the airways, though. Yes.
Great. And Dr. Richard Anthony asks, again, notes the images are truly stunning and great, great data. He asks, do you by any chance know how the pre-ribosomal RNA staining cells correlate with auramine Nile red and auramine negative Nile red positive staining MTB cells? As if they are the same or similar populations, this would be easier to implement in other centers. Any thoughts about the alternative staining techniques?
Yeah, so we do know that the general abundance of bacilli correlates with aramine rhodamine staining and also regular AFB staining. We have not added Nile red to that as far as I can remember. No, yeah, we have not evaluated Nile red in that setting, but with aramine rhodamine, we do see the same abundance of bacteria across the granulomas.
Do you have any thoughts or comments? Sorry, go ahead.
And how those populations, we haven't correlated those populations or the intensity of staining, for example, with the intensity of staining for the pre-ribosomal RNA or the 23S transcripts.
Very good. Okay.
The next question is for Margarita from Galo Goig. Hi, Margarita, wonderful presentation. Do you have any plans to test differences in transmission? I was wondering about the possibility of testing the capacity of transmission of mice infected with different strains, severe versus mild, to a naive population. Thank you.
So mice are not a good model to study transmission because they don't transmit the bacilli. So we can't look at direct transmission to study that in mice. What we can do, however, is to, because we know, I'll start a little bit before, we know what some of the determinants of the transmission, and we know that we need to have certain types of lesion, and that we can modulate in mice using the C3H FEJ mice that Joshua presented, because they form lesions that are more comparable to the human ones than the Black 6, and we are doing that. The other way we have to study transmission or to get a picture of transmission at molecular level using the mouse model is to look at specific immune signatures associated with strains that are not as I showed, severe or mild, but that are more transmissible in the population or less transmissible in the population, and see if we can figure out specific signatures, and particularly immune signatures associated with those differences. So it's we have to go around the issue. It's not as simple as study direct transmission from one mouse to the next, because that doesn't happen.
Thanks, Margarita.
Thank you, Margarita.
A follow-up question on that, Margarita. There's a query from Dr. Leopold. In addition, first of all, great talk. In addition to the MTB diversity impact you show on pathogenesis, there is the impact of host diversity in different regions. How do you envisage addressing this additional diversity for optimal understanding of the host pathogen interactions?
Yeah, so I think that's a very good question. And our strategy was to go deeper than the MTBC diversity. So that's why we chose clinical isolates that were from the same lineage and sublineage and studied their interaction with the host. And I think we'll get a lot of information and useful information if we start to stratify for both the host and the bacteria differences in the different studies. So of course, our study was performed in our geographic area, and there was a match between the bacteria and the hosts. But as I showed in the mouse model, completely different species, we have the same effect. So I think there are factors that the pathogen will explore, factors that will depend on specific host backgrounds, and factors that the host will completely determine. So we really need to start matching pathogens and hosts more often than just studying the host or the pathogen separately.
Yeah, that is interesting. And when factoring in, the variety, the sort of diversity of nutritional status for the host as well, probably compounds much of this as well, in tuberculosis in particular. A question for you, Josh, if I may. This is from Eric Nuremberger. Josh, thank you for the lovely talk. These are exciting results. One clarifying question I have: what was the duration of treatment with INH or RIF in the experiments looking at the RS ratio and morphological heterogeneity in Kramnik mice? And a second question is, to what extent do you think the differences in morphology post-treatment reflect the mechanism of action of isoniazid on mycolate synthesis, staining, rather than differential selection of pre-existing subpopulations? And have you looked at other drugs?
Yeah, thanks for the question. So to answer the clarifying question, the time of treatment was 2.5 weeks. And then the second question was the impact of INH on mycolate synthesis. So that question, we're still a bit unsure. The heterogeneity studies are really just now starting, and the two drugs that we've looked at are INH and RIF so far. So I would say that I would hesitate to comment too much more on the heterogeneity and the impacts of synthesis there at this point, but we'll see as the studies continue.
That sounds great, Josh. Thank you. I'm sure you're going to look at other drugs as well. This is exciting technology.
Yeah.
Well, with that, I'm going to thank the presenters once again, Professor Churchyard, Dr. Vasquez, Dr. Saraiva. That was a fantastic series of talks. We're going to switch over now to the oral abstract presentation session. I encourage you to continue to submit your questions in the chat, in the question box on the program platform, and we will at the end reassemble for panel discussion. So thank you very much all over the world for watching this plenary session.